From male gametogenesis, let's now shift our discussion to female gametogenesis. The process of female gametogenesis can vary across plant species, although 70% of flowering plants follow the polygonum type of development. This pattern of female gamete development was named after the genus of dicots in which it was first observed. The series of events that leads to the development of the egg cell occurs in the ovule, which is inside the ovary. This diagram shows you a more detailed look at the female reproductive organ of a flower. At the organ level, you already know that the carpel is the female reproductive part of the flower. Within the carpel, you have the ovary, and inside the ovary is the ovule. The ovule has an outer layer called the integument. That is the structure that you see right here. An opening within the integument, which encloses the ovule, is called the micropyle. This is where the pollen tube goes through to fertilize the egg. Another structure that you can see here is the funiculus. This is a short stalk that attaches the ovule to the ovary wall. Aside from the integument, micropyle, and funiculus, we also have a structure known as the nucellus. The nucellus provides the tissue for the origin and nurture of the female gametophyte, including the archisporial cell. What is the function of the archisporial cell? The archisporial cell is the cell that divides to give rise to the megaspore mother cell. Meiotic division of the megaspore mother cell will give rise to the female gametophyte. This will signal the start of megasporogenesis. Initially, the megaspore mother cell will undergo 2 rounds of meiosis or reduction division to produce 4 haploid cells. So we have here our megaspore mother cell or megasporocyte. After undergoing 2 rounds of meiosis, meiosis 1 and meiosis 2, we now have our 4 haploid cells. What are haploid cells again? Haploid cells are cells having a single set of unpaired chromosomes. So just like in male gametogenesis, this set of 4 haploid cells is called a tetrad. But unlike in male gametogenesis, during megasporogenesis, 3 of these haploid cells will degenerate and only one, the largest one, the megaspore, will undergo mitosis. The process that covers the division of the megaspore mother cell to the development of the megaspore is called megasporogenesis. What happens to the megaspore upon completion of megasporogenesis? The functional megaspore will then undergo 3 mitotic divisions, 1, 2, and 3. Through this process, 8 identical daughter nuclei will be produced. This will then be followed by cytokinesis, producing a mature embryo sac containing 7 cells. What are these cells? We have 1 egg cell, abbreviated as EC. We have 2 synergids, abbreviated as SC. We have here the antipodals, abbreviated as AC, and then the central cell, which is abbreviated as CC. Except for the central cell, which has 2 nuclei, the rest of the cells within the embryo sac has 1 nuclei each. The process that begins from the mitotic division of the megaspore up to the formation of the embryo sac with 7 different cells is called megagametogenesis. What are the functions of each of the 7 cells within the embryo sac? Let's start with the most important cell, the egg cell. The egg cell is the female gamete that will fuse with the male gamete or the sperm during fertilization to produce the diploid zygote. The zygote will then develop into the embryo. The egg cell is located at one end of the embryo sac, sandwiched between 2 synergids. The synergids are small, short-lived cells flanking the egg cell, as you can see here, and also in this figure right here. Synergids produce attractants that guide the pollen tube to the female gametophyte during fertilization. They also contain factors that control the arrest of pollen tube growth, pollen tube discharge, and gamete fusion. The synergids immediately degenerate after pollination. Across the polar end where the egg cell and synergids are located are the antipodals. So if you look at this figure here, you have here our egg and synergids, and across these cells we have our antipodals. An embryo sac will have 3 antipodal cells that are all haploid. The antipodals nourish the embryo sac and produce substances that help in the growth and development of the endosperm. Last but not least, we have the central cell. The central cell is the only diploid cell that is produced from the fusion of 2 polar nuclei near the center of the embryo sac. Aside from the egg cell, this is the only other cell that is involved in the fertilization process. During fertilization, the central cell fuses with one sperm to produce the endosperm. To summarize, the sexual reproduction of plants requires the development of gametes in the form of sperm and egg. Without the formation of both gametes, the plants will not be able to complete their life cycle. Gamete formation undergoes 2 distinct phases, sporogenesis and gametogenesis. In female gametogenesis, these 2 processes are specifically referred to as megasporogenesis and megagametogenesis in reference to the relatively bigger size of the egg cell relative to the sperm. The structure that produces the female gametes in flowering plants is the embryo sac. The embryo sac, which is produced within the ovule, will have 1 egg cell, 2 synergids, 3 antipodals, plus 1 central cell. Of the 7 cells, only the egg cell and the central nuclei will be fusing with 1 sperm each during fertilization.